Okay. A very good evening to all the attendees who have joined today in this special public lecture, which is being organized by the National Academy of Sciences India Delhi chapter and MHRD-Institution Innovation Council, Deen Dayal Upadhyaya College, University of Delhi, under the aegis of DBT Star College program. Today we have with us Professor Graham R. Fleming from the University of California Berkeley, and who will be talking to us about the molecular magic of the primary steps of photosynthesis. As a customary, I'll introduce Professor Fleming to all the attendees. Professor Fleming earned his Bachelor's of Science degree from the University of Bristol in 1971 and his Ph.D. in chemistry from the University of London in 1974. Following a postdoctoral fellowship at the University of Melbourne, Australia, he joined the faculty of the University of Chicago in 1979. There he rose through the academic ranks to become the Arthur Holly Compton Distinguished Service Professor, a post he held for 10 years starting in 1987. At University of Chicago, he also served for 3 years as the chair of the chemistry department. In that role, he led the creation of University of Chicago's first new research institute in more than 50 years, that is the Institute of Biophysical Dynamics. In 1997, he came to University of California Berkeley as a professor of chemistry, and he started and directed a new division of physical biosciences for Berkeley Lab. Throughout his administrative career, Professor Fleming has remained a highly active and successful scientific researcher. He has authored or co-authored more than 444 publications and is widely considered to be one of the world's foremost authorities on ultrafast processes. His ultimate goal is to develop artificial photosynthesis that would provide humanity with clean, efficient, and sustainable energy. He is a member of the National Academy of Sciences and the American National Science Academy, American Philosophical Society, and a Fellow of the Royal Society, the American Academy of Arts and Sciences, and a Foreign Fellow of the Indian National Science Academy. Professor Fleming was appointed Vice Chancellor for Research at the University of California in 2009. Through high-level positions at University of California Berkeley and Lawrence Berkeley National Laboratory, where he was Deputy Laboratory Director from 2004 to 2006, he has been involved in the formation and operation of multiple major initiatives at Berkeley and LBNL. These include the 500 million British pound funded Energy Biosciences Institute, the California Institute for Quantitative Bioscience, and the Simmons Institute for the Theory of Computing. With these words, I request Professor Fleming to kindly in his talk.
Thank you, Manoj, and good evening, everybody. Let me see if I can share my screen. How is that?
Yes, coming up.
Is that good?
Yeah, absolutely fine.
Fine. Good. So I changed my title slightly from the primary steps because I realized that my talk might be 2 hours if I was to do both the first 2 steps in photosynthesis. So I'm going to focus in this talk on just the way in which plants and other green photosynthetic organisms collect the sun's light and focus it on the reaction centers that initiate the chemistry. I thought I might start with this quote from my thesis advisor, Nobel Prize-winning photochemist George Porter, that the study of photosynthesis is and probably always will be the greatest odyssey of the photochemist. I don't think I realized almost 50 years ago when I was a student that I would still be thinking about photosynthesis all this time, after all this time. And although we've learned a lot, there are still a great many things to learn. But let me take a few steps away from the detail and start with the big picture. If you want to think about things on a long time scale and a large planetary scale, it's always good to talk to a paleontologist. And so this is a colleague of mine, Tony Barnowski's plot, and it's the mass of megafauna on the planet over the last 100,000 years. Now, the first thing you're going to ask me is, what are megafauna? Well, they're you and me and every animal that weighs more than 44 kilos or 100 pounds. So, as you see, for a very long period, roughly 200,000 years, the total number of these large animals on planet Earth was pretty much constant, roughly. And then something happened. Suddenly, in the late 1700s, early 1800s, the numbers started increasing exponentially. What happened? Well, in this period here, the Earth was, roughly speaking, in balance. The amount of energy coming in and being converted to food by photosynthesis was balancing the maximum number of animals that could eat it. And we concentrate only on these large animals because they either eat directly the vegetation or they eat other smaller animals that in the end eat the vegetation. So I would say that this long period here, 100,000 years or so, that's where we're in equilibrium. And now we've gone way out of equilibrium. How did we manage to do that? Well, we did that by using old photosynthesis. We call it fossil fuels. Yes, we mechanized farming. We invented fertilizers, which consume enormous amounts of energy to make them. But we've got now very far from equilibrium. And this is a problem because if we look, now this is interesting. My computer has frozen. If we look at global yields of the main food crops, they're increasing only slowly or indeed leveling out. This is a major issue because 2/3 of all the calories that are harvested come from only 4 crops. So that the planet is essentially dependent for food supply on 4 crops. And the projected demand for these crops is outstripping any reasonable projection of the yield of, as you see in this plot, of rice, wheat, or cassava. We can't sustain the increase in the mass of megafauna, the things that we eat by and large, which has increased by 10 times over the 300 years or so since the Industrial Revolution. And so we really need to start thinking how to increase the efficiency of photosynthesis, or to increase the yield of the crops that we eat. Of course, there are many different ways to do this, but as I'll conclude right at the end, one of the most important ways is in optimizing the response that all green things produce to fluctuations in the intensity level of the light that they receive. So to begin, we need to understand a little bit about what's going on with the absorption of sunlight and its collection and transfer to the components of the photosynthetic apparatus that do the chemistry. So we begin at the molecular level, where we're going to talk a little bit about the rates and mechanisms of electronic energy transfer. We're going to see that we need to understand quantum mechanics. We'll be talking about only a few molecules, and the timescales will range from very, very short, to tens of femtoseconds, up to tens of picoseconds, and the length scale, tens of angstroms to 30 angstroms. Then we're going to want to step up and start to think about larger structures. We cannot do the detailed quantum calculations as soon as we start to get hundreds or thousands of molecules in our picture. And so we're going to need to coarse grain. And then we're going to step up again. We're going to go to the whole membrane, or a piece of the whole membrane. And now we're going to step up in length to several hundred angstroms, maybe even microns. And again, we're going to have to coarse grain. And the successive coarse graining, as we go from the very detailed microscopic picture down here, this kind of a system, up to the membrane scale, it's my contention that that can only be done properly by having an accurate description of the level below the one that you're focusing on. So if you want to know about this membrane, you really have to understand about these subsidiary structures. If you want to understand them, you really have to know about the individual components. This is a classic description of what's called a multi-scale problem. And we have a massive multi-scale problem here, going from angstroms to microns and from femtoseconds to minutes and hours. If we look over here, what you see is actually the light level measured in the field in Sweden in August, I believe. And this is during the course of the day, so light increases at dawn, reaches a maximum and decreases. But what are all these ups and downs? These rapid fluctuations are clouds passing over where the measurement was done. And the plant has to respond. So that's the first thing. The light level in the middle of the day is too high and will cause damage. I'll come back to that. But if the plant has a mechanism to kind of switch off, turn up a dimmer switch, then when the light level suddenly goes back to normal levels, it's going to have to turn off this dimmer switch. Otherwise, it's not going to be working very well. And so the time scale on which this dimmer switch mechanism, which I'm going to talk a little bit about, is turned on and off, is one of the things we can try to optimize to improve crop yields. This happens all the time. It happens because leaves under leaves have fluctuating light because the wind is moving the canopy above them. I mentioned there are clouds. If there are aquatic organisms, algae living in the oceans, for example, where about half of global photosynthesis occurs, waves produce lensing, and the sunlight gets focused very strongly sometimes. And again, that's too much and will cause damage. And so the idea that a leaf is just some sort of green solar panel that's sitting there passively absorbing the sunlight is really absolutely incorrect. It's a highly dynamic system responding continuously to environmental changes, especially changes in how bright the light is. So how are we going to make a start on this massive multi-scale problem? Well, there are really four things that we need. we need to understand the structure, the structural biology. Now, this has been a major development over the last 30 years or so, because all the crucial components of membrane proteins are extremely difficult to crystallize. And so what big breakthroughs have occurred more recently in terms of cryo-electron microscopy. But earlier on, it was the use of synchrotrons, enabling very small crystals to have their structure determined. Then, of course, genetics and the ability to engineer plant genomes has come along, and so we have access to different kinds of mutations. And my own group has had a long collaboration with my colleague Chris Yogi in plant and microbial biology at Berkeley, and we'll use some of his talents a little later in the talk. Then, where we come in is understanding quantum dynamics. I'm not going to talk too much about the theories that we use, but we don't really have time, but simple ideas about energy transfer of hopping from one molecule to another do not work. Excitations are delocalized over several molecules, and this can be described by something that was again developed in my group, generalized Förster theory, or exactly by a method called the hierarchy equations of motion. And then we need ways of measuring, and this is where my group has been heavily involved in creating new kinds of multidimensional optical spectroscopies. And I'll talk a little bit about a very new one in a few minutes. All right, so let's just take a really big step away from all this detail and think what's going on. Well, we often talk about the major component, a piece of the leaf that makes it look green, as an antenna. Most of the chlorophyll is not involved in the chemistry of photosynthesis. Most of the chlorophyll is simply absorbing photons of sunlight and passing the energy that is created through optical excitation from one molecule to the other, to the next, until finally it gets to the reaction center. For each reaction center, there are several hundred, maybe typically 300 chlorophyll molecules that are funneling energy into the reaction center. So the analogy is with one of these radio dishes, where this big dish here is the collector that focuses the energy in the end onto the detector here, our reflector up here. So following the energy transfer, we have electron transfer, and we have worked on this quite a bit recently. I'm sorry, I won't have time to talk about that. So, okay, this energy transfer network through all these hundreds of chlorophylls. Of course, people have long thought about how this might work, and really the key idea came from Theodor Förster in Germany, transfer by inductive resonance. Very cleverly, brilliantly combined the electronic and nuclear factors necessary to calculate the rate in a formula that all of the factors are where all of the components that you need can be measured, and well, not very simply, I'd have to say, in case of photosynthesis. People used Förster's ideas to start to think about how fast the energy moved from chlorophyll to chlorophyll. In 1950, Arnold and Oppenheimer, the same Oppenheimer that was involved in the atomic bomb, or the Born-Oppenheimer approximation, also Professor at Berkeley, I might mention, came up with 10 picoseconds for the half time. 16 years later, Joyce's in the Netherlands came to the idea that the energy transfer was much, much faster, 100 femtoseconds. And at the same time, my postdoc advisor, Willis Robinson, suggested that, in fact, what he called exciton spreading could occur on the timescale of around 50 femtoseconds. He gave this in a talk, the only talk he ever gave on photosynthesis, and it's published in the Brookhaven Symposium, and the questions are also published. And clearly there was great skepticism in the 1960s when people mentioned timescales like 50 femtoseconds, that such a time would ever be measurable. So Prostein's a theorist who asked directly, how are you going to measure this? And Robinson said nobody has ever done anything on this timescale. Prostein suggested it might be very difficult experimentally to ever settle this question. This, of course, is an open invitation to people to try to make that not true. And so that's one of the things that we've been doing. So how are we going to measure these timescales? We're convinced that it is really, really short. You can easily make an estimate that Dyson's did by just taking a random walk with a number of chlorophylls and the quantum yields, which are very close to unity at low light levels. So we're going to have to make some very, very rapid measuring. Then we have the next problem. We don't see one species go down and another species come up, because most of the molecules are exactly the same. So how do we know where the energy is? Is it going to be just a simple rate process, or we can call it an exponential, or is it something more complicated than that? So these thoughts have made multiple people around the world think about new spectroscopic methods and new theoretical approaches. As I mentioned already, once we understand this at this microscopic level, we're going to have to figure out how to cross-grain. And then when we're satisfied with our cross-graining, we're going to have to combine all these ideas together with a biochemical model for how the feedback, this dimmer switch I've mentioned, how it works. Think about it as an air conditioning control system. There's a sensor and actuator and a feedback loop. That's exactly how plant must work in some fashion. It's an engineer, and it's a biochemical engineer. Okay, so the first method that I will talk about, just to get you calibrated slightly, is a method that we first applied to a photosynthetic system in 2005. It's two-dimensional electronic spectroscopy. And so what you have here converted to two frequency axes is an excitation frequency axis in wave numbers, and a detection frequency axis also in wave numbers. And you'll see over here big T equals 0. And this is what we call the waiting time. So along the diagonal axis of the 2D electronic spectrum are the peaks of the ordinary absorption spectrum. They're elongated along the diagonal by inhomogeneous broadening. I'm not going to talk too much more about that. It's not terribly well resolved. And then we see cross peaks. These cross peaks at T equals 0, waiting time of 0, only exist because there are electronic coupling mixing between the components of the system. This is a 7 or 8 bacterial chlorophyll protein. So we can analyze this spectrum and learn about the pathways of energy transfer by looking in detail at these spectra with a series of waiting times and seeing how these cross peaks evolve. We can learn how the energy moves, so at least rough approximation through the system. But when we make plots of the intensity of these peaks along the diagonal as a function of time, we got a big surprise. What we saw were these oscillations. And so you see here, for example, on this cross peak, and you see that it's going up and down in a regular kind of way, as is the main peak on the diagonal, in a complicated looking way. And it's like, well, maybe this means that the energy isn't moving like a ping pong ball bouncing around, but it's moving more like a wave, and what we're seeing is the oscillations of a wave. And we published a paper in 2007. This created an enormous level of interest. It's been cited around about 3,000 times so far. It's even produced the story for at least one novel. It's called Solar by Ian McEwan, famous novelist. And a great deal of discussion, lots and lots of theory, not much in the way of new types of experiment. I think what's causing these oscillations, a consensus is emerging that mixed electronic and vibrational states, or vibronic excitons, might play a key role. I'll come back to that in a second. But just for fun, let me show you a little bit about some of the controversy that has arisen here. So the theorists have had a field day here. They've been arguing with each other in the absence, really, of any new clear experiments. Some theorists say, well, it's not quantum mechanical. Others say, oh, well, it's just ground state vibrational wave packets. It has nothing to do with anything that involves photosynthesis, really. Still others say, well, it doesn't really matter anyway if you see this with your femtosecond laser, because photosynthesis happens in sunlight. And that's different somehow. And the same kinds of issues actually arise when people talk about charge separation in both the photosynthetic reaction center and in organic photovoltaics. My good friend and very creative theorist, Sean Mukamel, once said to me, one equation is worth 1,000 pictures. As you probably know, one picture is worth 1,000 words is a cliche. I think maybe one experiment is worth 1,000 equations. And so we start to think about what kind of experiment we might do. After spending 3 months in Oxford talking to people who do quantum computing, quantum information, I concluded there's really no unambiguous way to determine if a system is evolving quantum mechanically or not with ordinary light. Maybe we can talk very briefly at the end about ways of doing this with what's called quantum light, in other words, entangled pairs of photons, so that you can have access to the photon statistics. But if we want to understand what these vibrations are doing, maybe what we need is an experiment that looks at both the electronic states and the vibrational states together. And that led me to propose a new kind of two-dimensional experiment, two-dimensional electronic vibrational spectroscopy. I'll just talk about that in a second. But let me take a step back from this exciting discussion, which I have to say is not really arrived at a conclusion. Is it important? Do we really care if it's quantum mechanical or not? Is it just angels on the head of a pin kind of argument? I would say if we want to design a system ourselves that works as well as the natural system, yes, it's very important. Because if we don't know the design rules that nature is using, it seems to me we're unlikely to be able to reproduce its abilities. On the other hand, If we want to just understand how the whole membrane works, no, I don't think it's very important to understand this level of detail. It's sufficient to know that there are very rapidly communicating domains and slower steps, and that this thing at a cruder level can be described by this generalized first theory, which allows for delocalized states, but doesn't involve very complex quantum calculations. As I mentioned here, generalized first theory was developed by both independently by Sumi in Japan and Greg Scholes and myself in Berkeley. Okay, so I already showed you the two-dimensional electronic spectrum. We're now expanding the scales of the two axes. So this range here is going to go all the way from the infrared to the visible. So up here is a two-dimensional electronic spectroscopy that I showed you. Down here is two-dimensional infrared spectroscopy. My idea was, why can't we do this? Make a spectroscopy where the excitation is in the visible, but the detection is in the infrared. So the idea at its simplest is we would correlate the electronic and vibrational nuclear evolutions together. The experiment turns out to be considerably richer than that and somewhat more complicated to do than the simple picture shows you. But we don't have time to go through all of that. Let's say it's possible. And what does a two-dimensional electronic vibrational spectrum look like? This is a simple molecule in solution, crystal violet, and it undergoes twisting to a conical intersection and rapid relaxation back to its ground state. So just to get calibrated, here's the visible excitation axis. Here's the infrared detection axis, portion of it. These peaks that are red or yellow or green, they're just decreases in absorption of vibration frequencies on the ground electronic state. The blue peaks, they're new vibrations in the excited electronic state. And you see it gets stronger here by half a peak per second. And I just want to show you that by the time you get to 3 picoseconds, a new vibration has appeared. What we believe this to be is the molecule immediately after it's passed through the conical intersection. It's now in a region of the ground state potential that was not occupied previously. And it then relaxes back to the original starting position. So this maybe gives you a little flavor for how these spectra look, and a very simple look into what you can get out of them. But there's something else about these spectra which is, I think, unique. If we look at the spectral lines, they're not round. They have, we draw a line along the maximum of the feature, either with respect to the excitation axis or with respect to the detection axis. We find that there's a slope. We call it the center line slope. And I think you can see in these plots that this slope is positive. Positive and the same, actually, plotted in both dimensions. And it decays over time. In this simple case, it just decays with the vibrational dephasing time. We'll see much more complicated cases of this as we go on. This is a cross-correlation function. Similar slopes exist in degenerate two-dimensional spectroscopies, electronic or infrared, but they're autocorrelations. They're different. This is a cross-correlation between the electronic dipole moment and the vibrational dipole. Okay, let's get on to photosynthesis before I run out of time. So here is a cryo-EM structure at 3.2 angstroms of what's called the photosystem 2 LHC2 supercomplex. This is the photosystem 2 core, it's a dimer. This is where the electron transfer begins. This is a trimer of light harvesting molecules called light harvesting complex 2, LHC2, which we're going to focus on for a little bit. Each one of them contains 14 chlorophylls, 8 chlorophyll A and 6 chlorophyll B. And this is the main, there are multiple copies of this primer in the whole membrane. This is the main light collection unit that everything green uses. You see a satellite picture of the Earth, and you see regions of green. What you're actually looking at is this molecule. Half of the chlorophyll on the planet is in this LHC2 molecule. So it has, as I said, 14 chlorophylls in each one, monomer of the primer. The spectrum is very broad because these chlorophylls interact with each other. And just like when you mix two atomic orbitals, you mix two electronic excitations, you get two new states, we call them excitons. And they repel each other, one goes up and one goes down in energy. And by this means, and the fact that there are two kinds of chlorophylls, LHC2 covers a large chunk of the absorption spectrum. So I show you down here the absorption spectrum, the brown line of an LHC2 trimer. Since there are 14 chlorophylls in the monomer, there will be 14 excitons. And this is roughly what their energies are. And this broad thing is the spectrum of the laser that was originally used to make these studies. Okay, so now here is a 2D electronic vibrational spectrum of LHC2, quite recently published. And you see a whole series of remarkable things in this. First of all, if you look at this, the absorption spectrum, the brown line, yes, there are two peaks, but there's pretty filled in between. And then it just goes flat out here forever, more or less. But what we see in these spectra is all sorts of detail. Two clearly separated regions. This is the chlorophyll A absorption this time. This is the chlorophyll B absorption, and then these are higher vibronic bands. In an energy transfer system, there is no, it's not so simple that, for example, chlorophyll B has slightly higher energy than chlorophyll A, and so you would expect to see the chlorophyll B peaks go down and chlorophyll A peaks go up. To some extent that happens, but actually, in an energy transfer system, what happens is the spectrum becomes more and more symmetric. As you see, if you look at longer times now, it's looking rather similar on both sides of this BA line. I confess when we first saw these spectra a few years ago, we didn't really have a clue how we would go about analyzing them. But we did decide, let's try singular value decomposition. And we got these 4 singular values, singular vectors. And I'll just show you this. Here's Robinson's 50 femtoseconds. And it corresponds to both chlorophyll B to chlorophyll A transfer and relaxation within the chlorophyll A's. You see slower rates, you see bottlenecks, rates that go up and go down again. And you can make a good deal of detailed analysis from this. But it's still basically a kinetic picture. Is that really right? I mean, the analysis that we did initially was based on the simple idea that the vibrations are acting as flags. they're saying excitation is here or excitation is over there. Is that really right? Or vibrations mixing with the electronic states and things are more complicated? Yes, it turns out things are quite a bit more complicated. Vibronic coherences dominate at short times. These spectra become very sensitive to the vibrational electronic mixing. This is where the work of an Indian postdoc, Bachelor, comes in. And just want to give you a little flavor of how complicated things get. This is the simplest kind of model you could imagine, a dimer. And each component of the dimer has one vibration in its ground state and one vibration in its excited state. That gives you 6 vibronic excitons. It produces perfectly usable looking absorption spectrum. But now if I start to calculate rates, all hell is let loose when the vibration frequencies become resonant with some of the electronic states. And I don't want to go through this in great detail, but here are the 6 vibronic excitons. This is a non-resonant case. And this rapid relaxation here, this is what first the theory will tell you. It's just the upper exciton to the lower one. Nothing really complicated. Yes, there are faster rates up here too, but this you think of a simple system. What happens when you get resonance, in other words, energy matching between these levels, or near energy matching? There's all sorts of rates become very fast. You can have rates varying by a factor of 100. And so, does nature actually use this? Particle, after all, has an awful lot of vibrations. Does it use this in order to aid in the energy transfer? Well, let's now go back to our spectra and try to understand them a little bit more. One of the things we noticed in looking at these spectra is that kind of quartets of peaks appear. Remember, these blue things are the excited state bands. So these are vibrations of chlorophyll A and chlorophyll B that we're seeing here in the excited state. And if we look in this square, for example, we see a little set of levels. These tell us who the energy transfer partners are. And now I want to take you back to the slopes that I mentioned, these center-line slopes. We look at them, they have anti-correlated, the pairs have anti-correlated center-line slopes, and they have anti-correlated oscillations at the frequency of the donor acceptor energy gap. And so I'm going to now start to focus on these 2 quartets here. Now, just going through a model so things are very clear. Here is here are the center-line slopes for the peaks marked E and E prime. And you see they're just absolutely out of phase with each other. Same thing for C and C prime. We can Fourier transform these oscillations and we find this one frequency, and it's the energy gap between the vibronic excitons. And now if we plot the other way, we plot the excitation frequency versus the detection frequency. If we do that, then if I look at that 245 wave number frequency, it picks out for me in the spectrum the 2 electronic absorptions that are connected by that frequency. And so I can go through and I can do this with the real spectrum. And these oscillations are not so pretty because there are lots of frequencies present, but I can find, for example, a specific frequency. This is 650 wave numbers that I've marked here. And I can then go and look now, see which states are connected by the 650 wave number vibration. In other words, the state here and the state here. And we can now go back and identify what particles are involved there. And what we're seeing is that the vibrations are enabling LHC2 to have a very rapid energy transfer over quite large energy gaps, because the vibration is brought into service to cover the energy that needs to be disposed of. And we can build up an extremely detailed picture of how energy flows through the complex. Pallavi decided that she was going to go for broke, and she has developed, when she was a Ph.D. student at the Indian Institute of Science in Bangalore, a method that's quite efficient for calculating quantum dynamics. Not exact, but pretty good. And she put in 48 different vibrations for particle and calculated all of the rates in LHC2 monomer. And what you see here is when you don't have these resonant vibrations, you just get these green rates that are like up here, for example, here, that limit the number of pathways in the whole structure. As soon as we start putting all these vibrations in, then the vibrations open up a whole range of different pathways through the structure, giving it very, making it very robust and very efficient to transfer the energy. I think if I'm not going to run out of time, I'm going to need to move up and scale. So we've been focusing very narrowly down on very small things. Now let's go up. So here's a section of a chloroplast. There's regions of the membrane that are all squished together, called the grana, and regions of the membrane which are more widely spaced, called the stroma. If we make a cartoon of what this looks like, we've been focusing in here on these bright green LHC2s, looking now cross section. Photosystem 2 of the big things. You notice it sticks out of the membrane. The oxygen evolving complex sticks out of the membrane. We're going to use that in a minute. And these are physically separated from photosystem 1 and ATP synthase, which lives in the unstacked region. Here's an electron micrograph. Now, what we're seeing, these white spots you see here, these white spots are in fact the oxygen evolving complex sticking out of the membrane. This is the photosystem 2 supercomplex that we can localize because it sticks out of the membrane a little bit. And everywhere between is filled in with LHC2 trimers, the whole membrane. Sometimes the membrane forms these semi-crystalline looking arrays. We're just going to focus on this more irregular structure where everything is dispersed. So if we're going to go and get from this very detailed picture we have of the energy in the LHC2s to how this whole membrane behaves, one question we have is, how big a piece of the membrane do we need? And that is going to depend how far does the energy move. When a photon is absorbed, can it only visit one supercomplex, or if one supercomplex is busy, so to speak, it's already just transferred an electron, it's not ready to do it again, can the excitation go and find another one? And this is obviously a complicated question of competition between trapping, excitation motion, and speed. So we need obviously a model membrane, and my colleague Phil Geisen and his student Anna Schneider made this model a few years ago. This membrane is really quite remarkable. It's absolutely packed with protein. 60 to 80% of the membrane is protein. And then this picture, which looks like a cartoon, but it's actually very detailed. These blue pill-like things, these are the photosystem 2 supercomplex, and the green circles are LHC2 trimers. So this we're going to use as a basis for understanding how energy flows in the membrane. So I'm now jumping quite a lot of steps because I want to run out of time. We made a model of the supercomplex. We parameterized things so that we could deal with about 200 chlorophyll molecules, and it all seemed to work quite well. So then we decided, let's try membrane. So using that model for the membrane, we made up a 200 by 200 nanometer patch with about 30,000 chlorophyll molecules in it, and applied our model to predict what the fluorescence decay of the membrane would look like. And if it hadn't worked, I have no idea what we would have done, because there's really nothing to adjust. We didn't allow any kind of level of adjustment in parameters. This is not a fitting exercise. This is a prediction. And so this is the measurement in red spots, and the black line is what the simulation of that 200 by 200 nanometer patch membrane looks like. I don't want to go into great detail about this, but many people, when they measure the fluorescence, fit it with exponentials. I just want to show you the blue. There's two different time ranges, and just focus on the blue lines in these. These are the number of time constants in that dispersed membrane structure. The number of time constants in the range 0 to 300 picoseconds, and in the range up to a nanosecond that are present in this black line. Hundreds and hundreds and hundreds of timescales. You can make a kind of kinetic spectrum. That's this black line here. You could pretend you're an experimentalist and fit this, and you get 3 decay times, which actually don't really match with the kinetic spectrum at all, and their meaning is totally unclear. So this is just a comment on getting exponentials to very complicated underlying phenomena. Let's go a little bit further with this. My animation is not going to work today, right? Worked yesterday. It's not going to do it. What I would show you, if this was working, is when I thought the excitation on this particular LHC2 trimer, you'll see that it can visit multiple reaction centers. The diffusion length is characterized here in this plot, which is just the diffusion length is just how far the next station can move before its amplitude is decayed to 1/e of the initial value. It's 50 nanometers. That's enormous. If you look at the kinds of numbers that organic photovoltaics have, it's 10 times bigger than the natural system. That again speaks to why we might want to understand this. And so we have an excitation that can move quite far. That is, of course, great to make an efficient system, but it may also cause a problem. Let me just again appeal to you to see that we're actually have a model that's quite realistic. We went back to this old classic experiment with Joliot and Joliot in Paris in 1964. They looked at photochemical yield as a function of the number of reaction centers that were ready and able to operate. And we don't see a linear dependence, you see this parabolic dependence. And so that when half of them are closed, the yield is higher than 50% because an excitation can visit more than one reaction center. So that's the fundamental origin of the parabolic dependence. And our membrane model produces exactly this parabolic dependence. It also reproduces variations in the fluorescence lifetime if you close all the reaction centers. So this highly efficient system, why is it a problem? Well, as I already mentioned, plants under normal circumstances experience lots of different light levels. So we really have two contradictory requirements. I focused all on about moving the excitation hundreds of times, much faster than any dissipation. It's 50 to a few hundred femtoseconds time scale. But I'm going to have to also now add a new mechanism that when the light is too bright, I've got to get rid of the light, some of the light, in order to avoid damage. Because what happens actually here is if I can't do the photochemistry, some of the chlorophyll molecules will get into their triplet levels. And then, because we have this poisonous byproduct of water splitting, oxygen, the triplet chlorophyll will excite the triplet oxygen that's produced by water splitting up to its excited singlet state, which is one of the nastiest, most reactive chemicals that exists, and it will proceed to destroy the proteins. In fact, plants can be killed by too much light. And so we have to have, if we measure the rate of photosynthesis, it scales linearly with the amount of absorption, the intensity of light, and then it just turns over. And full sunlight is probably somewhere up here. So full sunlight in the middle of the day is just much too much. And we're going to have to figure out how plants dissipate this energy. Well, this isn't going to work. So what else might we do? There's a process that's got a strange name, in my opinion. It's called nonphotochemical quenching. It's kind of like a safety valve or a dimmer switch. And we know a few things about it. Let me sneak up on it. We know that it involves, first of all, let's ask it, how does the plant know the light's too bright? Well, senses the pH gradient across the membrane. There is a molecule that senses when pH drops inside the membrane, in other words, when protons are being transported across the membrane. There's also an enzyme that is also pH sensitive that converts one kind of carotenoid to another kind, and converts bioxanthin to zeaxanthin in excess light. By the way, we have the same problem in our eyes, and we have zeaxanthin in our eyes for the same purpose, because zeaxanthin will get rid of the triplet chlorophyll to avoid the possibility of getting a singlet oxygen. And we don't make zeaxanthin, you only get it from dark green vegetables. So your mother was probably right. So we know we have these two components, the pH sensing and the zeaxanthin formation. What's going on? Well, let's just go back to our highly detailed model. One thing we know is that the longest time an excitation spends anywhere is on a set of 3 chlorophylls, 610 to 612, and it's 2.8 picoseconds roughly. So this is how long we've got in order to catch the excitation. We don't have to be 100% efficient. We may have to have multiple goals to catch it. And that's what I mean by weak quenching. The quenching dissipation of the energy won't happen immediately one of the quenching sites is found. What we modeled in our work on the membrane now was adding in these quenching sites and seeing that you might think that the way to measure it, the way to understand it would be to calculate how many quenching sites there are and how fast do they quench. We found that that was actually not helpful because only the product of those two quantities was relevant. Because what is actually important is this diffusion line, which we call LD. And what we found is, for example, in the standard all groups study because of the genetics or Arabidopsis, this diffusion length drops from around 50 nanometers to about 25 nanometers when this non-photochemical quenching is turned on. So that's what the plant is doing. It's limiting how far the excitation can move. And how is it doing it? Well, I mentioned right at the very first slide that the question of photochemistry. So what mechanisms do we have available? Well, we have energy transfer. We could transfer, for example, maybe the zeaxanthin accepts the energy from the chlorophyll and then rapidly relaxes down to its ground state. A number of years ago, I asked my colleague Martin Head Gordon what happened if we made a dimer of zeaxanthin and chlorophyll. And he came back and said the lowest energy excited state is actually with a charge where an electron has jumped from the carotenoid to the chlorophyll. And so it's a charge transfer quenching mechanism. So these what are going on. Is that how it works? Measuring fluorescence in highly scattering samples is not too difficult. Measuring transient absorption, on the other hand, is a heck of a lot more difficult. However, our collaborator, Chris Yogi, started to study this very small microalga called Nonphotochemical. And we realized this is what we need. It's so small it doesn't scatter too badly, and we can do time resolved absorption experiments. Also, with genetics can be manipulated. And so we set out to see, can we find either the energy transfer or the charge transfer quenching mechanisms. And so we've got 3 mutants here. Normal alga. So this is very weak light. No non-photochemical quenching. Turn on a bright light. Fluorescence lifetime drops dramatically. We turn the light off and it slowly comes back. Not yet, even after 15 minutes all the way. If we look at what's going on biochemically in the wild type, we see zeaxanthin being progressively made when the light is turned on and slowly going away. If we take a mutant that is not able to make the zeaxanthin, like the green spots, then there's a very minimal response to the bright light. Similarly, if we have a mutant that can't sense the pH change, very little response to bright light. This is just a different way of plotting the response. If we look at the biochemistry, we're still making the zeaxanthin when we don't sense the pH. That's this one, but we don't really see the quenching. And here we don't make any zeaxanthin at all. Okay, so what happens when we now? These are experiments where we look where we would expect to see the carotenoid singlet, right? And we indeed see it when the bright light is turned on. And in this experiment, we look at the wavelength where we expect to see the zeaxanthin radical cation, and indeed we see it when the bright light is on. So both of those are operating in at least in microalgae, and I strongly suspect in every green system on Earth. And so I think we've done this now in live cells here, the mutants, where we just don't see these absorptions. In either case, the ones without the pH sensing or the one without the ability to make the zeaxanthin. In neither case do we see the excited singlet carotenoid or the radical cation. So I think I've taken too much of your time. Let me try to sum up. All green photosynthetic systems control the efficiency of their light harvesting. The range of excitation in photosystem Q membrane is 50 nanometers about. If we don't have any of the quenches turned on. And to understand what's going on, you have to include properly the competition between reactions and quenching sites. You need to use this delocalized Berkeley theory. And we really need to include in our picture timescales from tens of femtoseconds up to an hour. Okay, I'll just finish with this very last point here. I showed you in this last part, when you turn off the bright light, the response doesn't recover very quickly. And by just speeding up the response a little bit, my colleague Chris Yogi and Steve Wong in Illinois, in a real field of macro plants, showed you could get 15% increase in the yield by just increasing the speed of the turn off a little bit. So there's real promise once you understand these mechanisms and the genetics behind them to optimize. So we did all this. The 2D EV experiments were done by Eric Arsenault and UK graduate student, UK UNEDA, and Japanese postdoc. The calculations, I mentioned Pilates. The modeling of the membrane was done by Doran Bennett and Kapil Amanath, both now, well, he's still a postdoc, but he's now a professor. The measurements on the intact organisms were done by Korean postdocs in Park and the current graduate student Colin Deen, and these are my long-time collaborators. And thank you so much for listening.
Thank you very much, Professor Fleming. How this 1 hour has passed, I mean, we just cannot imagine, right? Starting from quantum mechanics, photosynthesis, botany, chemistry, everything has been merged together. So this shows that we really have to learn. We really have to learn a lot when we are talking about multidisciplinary research activities in that case. And there are a few questions, 4 to 5 questions. So I'll pick up them. The first one is, quantum coherence is a very fragile topic. What do you think is the mystery behind observing such effects in biological systems which are thermally quite agitated?
Yes. The phrase that physicists like to use is warm, wet, and noisy. Yes, they're fragile. And so in my view, they won't be relevant in anything that's slow. However, as I showed you, some of the key processes here are incredibly fast. And so I think that some effects of quantum coherence on these very fast timescales are quite conceivable. As I mentioned just very briefly, however, proving that something is evolving truly quantum mechanically and cannot be described classically turns out to be very, very difficult. This goes back to arguments of Einstein's about the fundamentals of quantum mechanics and this whole business of Bell's inequalities. I mean, which are fundamentally a statistical argument, and that is what you have to make to prove this. These are experiments we're starting to do now, but they're incredibly difficult, and I have no results to show you yet.
Okay.
But I'm trying to answer that question.
Okay. I'll take up the next one. What is so special about sunlight? Because if only excitation is what's required, then we can as well use X-rays, but that doesn't work. Why can't the energy reach the reaction centers in the absence of chlorophyll?
Well, we can, of course, we can excite the reaction center directly, but then it would be very inefficient. The sun is a low intensity light source. If you look at the cross section of a single chlorophyll molecule, it's at full sunlight, it would not get excited. And you have to compare that. If you're going to optimize the rate of photosynthesis, what you need to compare is the rate of absorbing a photon with the rate of the biochemistry. Biochemistry can run at several hundred times per second. But if you just have to excite one chlorophyll, it's probably going to only be excited even with bright sunlight once per second. So you're going to be 0.5% efficient right at the start. And so we want to be able to have excitation that runs, that enables the system to run optimally. So it's just the same as the radio telescope. Yes, the receiver in the radio telescope would work, but it would be terribly inefficient if it didn't have a large dish, the antenna.
Okay. I'll take up the next one. What is the different light absorbing pigments present in a leaf of a given plant? These pigments absorb light in different wavelengths, and different wavelengths are shown to have different quantum yields.
Yes, but if we switch to the language of solid-state physics, there's a band gap to transfer the electron to the acceptor. And so all the light that's absorbed above the band gap just produces heat as it relaxes down to the band gap. So yes, there are absorptions in the blue, for example, and in very bright sunlight, green light, somewhat surprisingly, is also an important driver for photosynthesis. But the extra energy of those photons below, above the band gap, in other words, above the energy of the first excited state of chlorophyll A, is just dissipated as heat. So it can improve the total collection of sunlight, yes, but it doesn't enable more energy to be converted.
Okay. So the next one is, will these vibronic mixing help increasing the energy transfer during photosynthesis?
Yes, well, we certainly believe that the vibronic mixing speeds up the energy transfer, enables multiple pathways to operate very efficiently as the energy moves through all of these photosynthetic complexes towards the reaction center. So yes, it's our contention that the interpretation of our experiments is that vibronic mixing is actually crucial to the speed. Just maybe take a step back. As soon as the photon is absorbed, you have to think a clock starts. And you have only so much time for that excitation to get through the reaction cycle. Because there is an excited state lifetime, and it will cut off the possibility of using that energy, the energy of that photon. So you have a couple of nanoseconds to get to move the energy hundreds of times. And if you don't, and if you want to yield at say 95%, you have to move the energy really, really fast. That's and more or less isotropically in the plane of the membrane.
Okay. I'll take up the next one. Does the non-perturbative hierarchical equations of motion, that is HEOM, approach is an effective method in simulating the EET dynamics and spectroscopic signals of photosynthetic light harvesting complexes?
Yes, it's an effective method, but only for small systems, because it scales very strongly with system size. And that's why, I mean, the calculation, including all those 48 vibrations, I think would not, even in the very biggest supercomputer, would not be able to do that using the HEOM method. This was first applied to energy transfer by Akihito Ishizaki when he was postdoc, but the idea, the hierarchy equations were developed by Yoshitaka Tanimura when he was Kubo's graduate student, Kubo's last graduate student. But I think it hadn't really been developed to the point where it could be applied to energy transfer until Ishizaki did it, and it's formally exact. Unlike all formally exact theories in science, that means you can't always use it. It's like the Schrodinger equation is exact, but it doesn't help when you want to understand something very complicated.
Okay. I'll take up the last three questions now. Since LHC comprises 3D configuration, isn't it will be more beneficial in understanding the insights of these complexes with 3D electronic spectroscopy than using the 2D electronic spectroscopy?
Well, maybe there are new insights. I think, however, if you have Gaussian statistics in your system, you can show, and this is in Hucknall's book, you can show that, in fact, there is no new information beyond the forward, the two-dimensional case. I think it contains now. System may not be Gaussian, and so it's possible. Yes. I think, however, there's still plenty to do in two-dimensional experiments.
Okay.
I think it would be very interesting to look and see if there are new things in 3D experiments.
Okay. I'll take up the second last one. There are two parts in it. The first one is there a possibility to have more or all of NPQ than fluorescence? And the second part is any clue to speed up the off-on time for NPQ?
Yes. Okay, let me do the first part. This is actually very interesting, but it doesn't apply in India, I'm fairly sure. Or maybe I don't know if there are evergreens in the Himalayas. But certainly in the northern and Arctic regions, there are trees, evergreens, that stay green all winter. But they certainly are not doing photosynthesis because they're completely frozen. And so they are permanently doing huge amounts of NPQ. They turn it on and leave it on all winter. And I know a group in Sweden who are working to understand exactly how this is done. Just to give you an example of the subtlety with which plants can turn on NPQ, if you take a peach tree and you pick the peaches, the tree responds by turning on the NP2 because it realizes it doesn't need to produce as much food now. It no longer has the fruit. So, as I said, the idea that these are just sort of passive objects is crazy. They're very, very at least as sophisticated as us, I would say, in responses.
Okay, okay. So I'll take up the last one now. Could you please elaborate how the mutant with the pH sensor inhibited is acting as a control in an experiment where the parameter that's being changed is the light intensity?
Yeah, so it's not, it lacks the control is we believe the pH is how the light intensity is sensed by the alga or by plants for that matter, slightly different protein. So it's not able to sense the light intensity, at least in the usual way. It probably suffers some damage. You saw that the fluorescence intensity, the fluorescence lifetime went down a bit when I turned the light on. Maybe that's just some damage and it doesn't recover from completely, but it doesn't have much of a response because it doesn't really know, doesn't sense that the light's too bright. It's like a thermostat where the thermometer is broken. So the thermostat doesn't sense that the temperature has gone up.
Okay, okay, okay. So this one last question has just come up. I'll ask this one, and that's end of that question answer session. Now that field is reaching to a general consensus of vibronic energy transfer model, what do you think of the role of correlated fluctuations of electronic energy levels between donor and acceptors in energy transfer?
Yeah, that's a very interesting question. And people have tried to look at this, and my good friend, there is a paper that it's a collaborator on by Shinji Saito at IMS in Japan, Institute of Molecular Science, where he did quite sophisticated molecular dynamics and quantum chemical calculations and looked at a little bit of this. Just in the FMO complex, it seems to be not so important, but I think this is very, I would say, if you had these large scale correlated fluctuations, yes, that would help to preserve coherence, and that's something that could be engineered into systems. I think it was an interesting idea. How good the experimental support for it is at this stage, or even theoretical support, I don't know. There have been quite a few simulations that seem to have produced variable results. And so I think it's an open question, but a very interesting one. And a very good place to stop, I think.
That's okay. That's okay. Thank you. Thank you very much, Professor Fleming. It must be, I think, around 10 o'clock there.
Oh, no, just 20 past 9.
Yeah, 20 past 9. Okay, fine.
Yeah, so
Yeah.
it's getting a little
10
late.
p.m.
Yes, it's getting
It's 10 p.m. over here itself. And I must say that all throughout your talk, more than 200, 225 attendees were there all throughout, and they've been immensely benefited by your talk. And the level of the questions which your talk has actually motivated the attendees to ask to, that really shows that now we have a good new dimension in which people can start collaborating and they can work in the multidisciplinary or interdisciplinary environment in that case. So on behalf of the National Academy of Sciences NDR Delhi chapter and the Deen Upadhyaya College, I would like to thank you for sparing your valuable time. It has been more than 90 minutes, and I'll end the session. But before that, any concluding remarks from your side?
Well, I've enjoyed the possibility to reach a new audience. It's certainly the longest distance lecture I've ever given. And I suppose this is our new world. And I know that the coronavirus is also serious in India and here. And so I think I'll just close by wishing everyone in attendance to stay safe. take appropriate precautions, wear a mask, and I hope I will see you in person.
Definitely. We
Future.
would love to host you maybe next year if everything goes fine, then in India and New Delhi itself. Thank you.
Thank
Thank
you.
you very much, Professor Fleming.
Okay.
Okay, so I'm closing this session, and
Yeah.
I request all the attendees to kindly do fill up the feedback form before leaving. Thank you very much. Thank you very much, Professor Fleming. Thank you.
Bye bye.
Okay, bye-bye.